Hello, everyone. This is yet another wonderful occasion of the inspiring online biological sciences seminar series of TÜBİTAK Research Institute for Fundamental Sciences, which we organize for national and international audiences, relying on the unifying feature of science in a unique intellectual space. Geographic boundaries, languages, and cultures are different. Science is one. With this in mind, it is my distinct pleasure to welcome all of you for this special event. Those of you joining us through the Zoom platform and those of you watching us through all social media platforms of TÜBİTAK, the Scientific and Research Council of Turkey. I am also very much pleased to introduce you our tonight's speakers, Dr. José Ordovas-Montañes from Harvard University, who is going to give a great talk entitled Single-Cell Biology of Barrier Tissues and COVID-19. Dr. José Ordovas-Montañes is an assistant professor at the School of Medicine, Harvard University, a principal investigator at Boston Children's Hospital, associate member at the Broad Institute, and principal faculty at Harvard Stem Cell Institute. He is also a Robertson investigator of the New York Stem Cell Foundation. Professor Ordovas-Montañes received his Bachelor of Science in Biology from Tufts University and his PhD from Harvard University, where he studied how the nervous system and the immune system function together as the principal sensory interfaces between the internal and external environments. Later, as a Diamond Runyon postdoctoral fellow at MIT, the Broad Institute, and the Ragen Institute, he began charting maps of human gut and airway and discovered how human stem cells can be shaped by and remember inflammation. Professor Ordovas-Montañes started his group in 2019 at Boston Children's Hospital, where his group seeks to understand the principles of how inflammation derives memory formation in human body tissues in order to program and reprogram them in human disease. With this, I want to thank once again Professor Ordovas-Montañes for joining us for this seminar. We are really looking forward to it. Over to you, Jose. You're welcome.
Thank you. Thank you very much for the kind introduction. It's a pleasure to be here this afternoon in Boston and this evening with all of you in Turkey as well. So I really appreciate the opportunity to share some of our recent work here into how we're thinking about the single cell biology of barrier tissues, and also more specifically thinking about this concept in COVID-19, which continues to be a ever-present problem that all of us are continuing to face, either both scientifically or personally as well. And so I'll try to give an overview, a general overview of how we think about this in the laboratory, and then go into a few more specific vignettes as well, and hopefully with some time for any interested questions at the end. So in our lab, we generally think about the idea of barrier tissue adaptation. And so barrier tissues are those like the skin, the gut, or the airway. And these tissues are composed primarily of cells like epithelial cells, stromal cells, neurons, and hematopoietic cells, and of course, many subsets and states within those. And what's quite fascinating about these tissues is that they're constantly dealing with distinct inputs, whether they be inputs from the outside world like microbes, abiotic factors, or injuries, and coordinating the appropriate outputs of host defense, metabolism, or repair. Now, if we think about this kind of traditionally, we might associate a microbial input with immune cells and host defense, but as technologies have allowed us to ask these questions more comprehensively, we've started to understand that these cell types and subsets distribute some of these tasks amongst them in order to coordinate the appropriate levels of each of these different outputs. And so within the lab, we become very interested in this kind of epithelial and stromal niche as a key driver of overall health within these tissues. And so this is largely inspired actually by my roots as an immunologist. And so here, this is illustrating one particular example of how adaptive immune memory in a barrier tissue can actually fundamentally change the response properties of that tissue. So, if I were to tell you that just having one or two cells within a tissue like, the airway or the skin would completely alter our response to a virus, you might be thinking to yourself, how could that be possible? But recent work over the past decade has highlighted that specialized subsets of immune cells, again, just one or two of them, maybe even within these tissues, can completely alter our ability from a tissue being overcome by viral infection, as illustrated here, to one that is completely protected. And here's some of the references for those of you who might be interested in learning more about this topic. But kind of overall, the idea that I'm trying to illustrate here is that the ability of the immune system, and also maybe more broadly tissues and these barrier tissues, to form either naturally acquired immunity or immunity that is programmed by a vaccine, can be greatly beneficial to an organism. And conversely, that some of these mechanisms may also be underlying inflammatory disease biology. And so within the lab, kind of inspired by some of these thoughts, we've been developing a broader view of how cytokines, which are these key signaling molecules of the immune system, can shape cells and cell states in these tissues. And so again, as an immunologist, you might associate memory quite specifically with examples of antigen-specific immunological memory mediated by antibodies and B cells and T cells. And this is, of course, an exquisite example of one type of memory. But at the same time, if you were to ask a developmental biologist what memory means to them, they might tell you about fate commitment and the stability of how a epithelial stem cell differentiates to its particular daughter cell, and the irreversibility or potential irreversibility of this arrow. And then if you ask a neuroscientist what they think about memory, they might tell you how episodic memory works, right? And how the circuits present within different areas of the brain are critical for storing and retrieving this type of information. And so thinking a little bit about how memory means different things to different fields, we wanted to ask, well, is it possible that inflammation could be remembered by cells beyond those outside of the immune system? And so primarily thinking about the epithelial, stem, and stromal cells, which are very long-lasting cells that compose these barrier tissues. And so in order to do this, of course, we can't rely on the specific elements of the B cell or T cell receptor as one might normally measure. And so we have to think, again, a little bit outside the box, perhaps. And so we started to think, what would memory mean in terms of a gene expression response? And here I'm illustrating in pink what either a single gene or a gene module might behave like in terms of its difference in the baseline or sensitivity or rapidity or maximum of its response in after stimulation with a particular agent. And so, as I mentioned, we're interested not only in terms of how immune cells contribute to this inflammatory memory, but also how epithelial or stromal or maybe even neuronal cells, again illustrated here in pink in this graph, in addition to being key cell types that contribute to tissue maintenance, are potentially involved in this process. And so, of course, inflammation doesn't just come in one overall type. There's many types of inflammation. I'll highlight when some of those are important throughout the presentation. And here I'll just introduce a couple of them. Namely, we'll be talking about type 1 and type 2 immunity today. And I'll mention which ones those are as I go through. And then there's also a type 17 inflammation. And so in the lab, what we study is the way that each of these 4 principal cell types, either within the airway or the intestine or the skin, are both responding to and driving these inflammatory processes. And so today what I'd like to do is share with all of you a few of our recent published works and then also some of our unpublished work on COVID-19. And so what I'll do is I'll discuss in a couple of brief vignettes the power of individual cells, so namely single cell RNA sequencing, to understand these diverse tissue communities, sharing some of our work on allergic inflammatory memory and human epithelial stem cells, and also some of our early work in COVID-19, where we were trying to understand what are the possible cells that are targeted by SARS-CoV-2. And then I'll spend the remainder of the time looking at actual samples taken from individuals with COVID-19 infection. And here we've been studying how local intrinsic immunity within the nose is able to maybe be an early barometer or dictator of overall disease trajectory. And this is all unified by understanding the question of which cell subsets and states in our tissue are really influencing disease trajectory. And so one of the challenges of studying these tissues as single cells is, of course, that they are densely interconnected and challenging to separate. And so we typically, as immunologists, rely on a series of cell surface proteins that might allow us to effectively stain these cells. And we separate them out using a technique known as flow cytometry, a very simple example shown here. And if we were to take this, what's known as gate, which then goes on to allow us to describe much finer granularity, we can then tell you about T cells and B cells and CD4 and CD8 and different types of CD4s using known surface markers. If we want to do the same thing for epithelial cells, that actually becomes much more challenging to do. And so one of the really fundamentally enabling technologies that allowed me to ask how are these barrier tissues being shaped by inflammatory diseases was the advent of single cell RNA sequencing. And so single cell RNA sequencing, even though now it might seem like it's showing up in almost every biological study that comes out these days, is actually not a terribly old technique. And it is something that is scaled tremendously in a short period of time. So some of the early studies involved laboriously mouth pipetting cells into single reactions. And then a few years later, we could start to use complex cell sorters to be able to sort individual cells and then process them with relatively slow and cumbersome protocols. But then just 5 or 6 years ago, some of these high throughput methods, originally things like DropSeq or InDrop, started to come online. And then eventually in my postdoctoral lab, my mentor, Alex Shalik, was one of the early developers of a very accessible technique known as seqWell, which we started to apply to ask questions in human inflammatory diseases. And now at many different laboratories, there's instruments such as this, which is the 10x genomics controller, or others from other companies. And what this slide is simply showing is that we've gone from being able to identify cell states to really charting entire maps of tissues. And that, of course, raises certain analytical challenges that we now need to overcome in order to make sense of this large amount of data. And so I won't get into all of the analytical methods that we apply in the lab, but what I'll do is I'll highlight this and tie it back to actually some of the biological underpinnings as to why single cell RNA sequencing works. So of course you might be wondering, well, single cells only have a finite amount of RNA, and some of the best techniques are able to capture 10 or 15%. And if I'm measuring 10 or 15% of what's within a single cell, how can I really reliably know who that cell is, or what it's trying to do, or who it might be becoming within a particular tissue? And so the reason that sampling a small fraction of the overall transcripts actually allows us to get reliable information is because transcription is organized into these modular units. And so through combinations of cell type specifying transcription factors or subset specifying transcription factors, and those that are responsible for states that are present in tissues or different types of inflammation, we can effectively sample, bits and pieces of each of these modules in different cells and start to group them together based on those genes and modules that tend to move together with each other in each of the inferred cells that we're really measuring by these types of techniques. And so to lay out what we do experimentally on one slide, We start off often from clinical samples. So here showing a small clinical biopsy bite from human intestine. And then what we do is we take these samples and we dissociate and encapsulate them using a variety of different enzymatic techniques. And then we sequence and align that data and we derive a cell by gene matrix where there is some structure, but we need to find it using techniques like dimensionality reduction and clustering in order to arrive at our illustration, which is shown here, where every point is a single cell in this two-dimensional space, which is capturing many more dimensions that we can't necessarily see. And then typically those points which are closer to each other and colored like each other are more similar to each other. And so what we need to do is identify some of those marker genes that allow us to give cells a particular name and to understand what it might be doing. And so we also try to collect other information from these tissues so that we can go back and, this isn't the be all and end all, this isn't the end state of what we're measuring, in order to generate these useful maps and then test and refine hypotheses on the cellular basis of disease. And so with that, by way of introduction, what I now wanted to do is share some of our work from about 2 years ago on how the human airway epithelial barrier is adapting during allergic inflammation. And then I'll connect this into what we've been learning about COVID-19 over the next 2 vignettes. And so here we have a representation of that barrier tissue that I was showing you. We're able to sample epithelial cells, stromal cells, and hematopoietic cells, unfortunately not neurons in these types of studies. And the main question that I had going into this work is how is the tissue parenchyma, so primarily the epithelial cells in these tissues, responding to immune module activation? And so what I mean by that is primarily type 2 allergic inflammation, which is synonymous with cytokines like TSLP or IL-33, which induce immune effector cytokines like IL-4 or IL-13. Now, some of the specifics aren't necessarily important here, but the broad idea that this disease is driven by type 2 or allergic inflammation is important. And so the specific disease case that we were studying was a form of allergic inflammation known as chronic rhinosinusitis, which manifests within the upper airway of a large chunk, actually, of the population. And so the way that this differs is that in a healthy individual, after challenge, the tissue might reset to baseline, but these individuals suffer from chronic and recurrent inflammation that does not go away. And so here we can illustrate this. In green will be the non-polyp samples, and in purple will be the more severe polyp form of the disease over the next couple of slides. And the main hypothesis that I wanted to test was that epithelial stem and progenitor cells, those long-lasting epithelial cells that give rise to the rest of the tissue, might be responsible for holding on to potential memories of disease that, no matter how hard you try to push the tissue back down, may end up getting stuck at a particular point. So we wanted to understand if this was happening beyond conventional immune cells within these tissues. And so this was nowadays a fairly small study of only 12 samples and 18,000 single cells, where we sampled across the disease spectrum of non-polyp to polyp, again, green to purple. We profiled this using the seqWell single-cell RNA-seq platform and went through those illustrative analytics steps that I showed you on the last slide. Now, needless to say, these types of studies don't happen in isolation, and really working together with a close friend and colleague, Dan Dwyer, who's recently started his laboratory at Brigham and Women's, and a variety of the folks over there, partnered together with the folks at MIT that really helped make the seqWell platform happen, individuals at the Broad Institute, in particular Sam, who was a graduate student, also now a postdoctoral fellow in my lab, together with me, who really taught me a lot of what I know about analyzing single cell data. And so together with this team, I was able to go in and generate this image. And now, of course, the question becomes, which cells are we finding, and what does it actually mean for the disease? And so here, what I'm illustrating now and kind of coloring by overall cell types, and blues are the epithelial cells, and pinks and reds are the immune cells, and in greens, we have the stromal cells. And what you can immediately appreciate is that grouping the non-polyp patients together and looking at the frequency of the total cells captured, we can see that they're fairly reliably sampling epithelial cells, stromal cells, and immune cells in each of these patients. Same is true for polyp individuals, but you can already immediately understand that there are some significant changes to the distribution of these cells. And so I won't spend time going into the different kind of pinks and greens on this, but rather I'll focus on the blue epithelial cells for the next two slides and then transition over to the work in COVID. And so the first thing that we wanted to do was to understand, what is actually, what do some of these changes in composition actually mean for the tissue? And so is it that we are getting larger amounts of all of the types of cells, or are we giving up some and instead gaining more of others? And so we applied a very simple ecological index of diversity known as Simpson's in order to understand that in non-polyp individuals that have a balanced or a more balanced epithelial barrier composed of stem cells known as basal cells and secretory cells and glandular cells and ciliated cells that help sweep away mucus and viruses and bacteria. This is now something that was almost completely lost. And now we have really an outgrowing of basal cells, so something that's known as basal cell hyperplasia, together with incomplete differentiation of secretory cells. And we can quantify this and we can look for significant differences between clusters. And we can also look at the overall impact on the tissue shown here as a significant decrease in this index of diversity. And now this nicely correlates with the rank-ordered clinical pathological evaluation. And since we were working here with a dissociated tissue where we're breaking the tissue apart in order to capture single cells, we also wanted to understand if we immediately take the tissue, snap freeze it, and capture it with bulk RNA sequencing, can we actually see changes that recapitulate what we're seeing in single cell data? And the answer is yes. And we can see this nice kind of secretory and glandular signal in the healthier individuals here in yellow, going towards the basal dominated signature up here in yellow for the individuals with more severe polyposis. And so how does this allergic inflammation influence cell states in the human airway epithelium? Well, what we did is we kind of went back first to where these epithelial cells come from, which is the stem cell compartment known as the basal cells in the airway epithelium. We can see here that in our cohort, just as individuals have appreciated histologically for decades, we see this thickening of the basal cells where typically there should be one, and now there's maybe 5, 6, or 7 in different regions here. So we can capture that within our single cell data, but we can go a little bit further than that. We can actually ask, even though these are staining the same red for a particular transcription factor, are they really just more of the same cells? And the answer is no, because these are cells that are trying to productively differentiate in polyps. So these clusters are our stem cells, and they're trying to reach across from left, which is kind of their starting point of this differentiation race, towards the right, where they are becoming more differentiated. And we can see that they're getting stuck here in the middle around 0.5. And instead, what we see in the non-polyp case is that these cells can actually go out and they're reaching out and they're productively differentiating, and they're giving rise to secretory cells. So the overall tissue is decreasing diversity and kind of getting stuck in this differentiation trajectory. And so this to us helped to provide an additional layer of explanation for tissue remodeling. And in work that I won't show here, but is available in the published paper, we went on to test this hypothesis in vitro that inflammation can actually be stored as stem cell memories within polyp basal cells. But what I want to highlight here and how it links over to COVID is that what I've shown you so far is single cell data from non-polyp and from polyp individuals. So even though this is milder and this is more severe from a clinical vantage point, both of these are still diseased tissue. So lingering in our mind, we wanted to understand, what if we could sample healthy tissue from a very similar location within the nasal mucosa? So what we did here is we effectively just took a toothpick, I guess the professional version of a toothpick, and then we scraped the inside of the nose of a few control individuals, healthy individuals, and then we compared the single cell profiles here, each single cell being a vertical column and each gene being a row, and we could derive a conserved core epithelial signature shown here, and then a healthy signature, which can be seen in the healthy epithelial cells. And then as you go towards disease, you see that health is lost and that these gene signatures are being turned on and that it's most severe within the polyp. Now, if we look not just at the individual genes, but rather what the upstream drivers of those genes might be, we find recurrent signatures in healthy individuals and a type 1 interferon antiviral driven environment, even at baseline. And that completely trades off towards here in purple an IL-4, IL-13. So this type 2 allergic driven environment in individuals with nasal polyposis. And so we, of course, at the start of the COVID pandemic, started to think, well, if there's a variety of individuals with chronic rhinosinusitis that have a very high type 2 cytokine driven environment, what does that mean for their antiviral type 1 response, right? So what are the implications for antiviral immunity in these patients, or as patients have distinct shifts in their epithelial state in this barrier tissue? And so, of course, when this pandemic came out, we started to realize that this was likely to be a virus that was transmitted via the respiratory route, given its primary lung involvement, though we've also appreciated that the intestine can also be a site of symptoms, and that there can be very disseminated and multi-organ involvement depending on the types of disease course that manifest within each individual. But we focus primarily on using our single cell atlases of the nose, of lungs, and of the intestine to ask what are the potential target cells of this virus. Now, of course, this is a very biased view of this disease. There's many aspects of COVID-19, ranging from social to epidemiological to the basically single molecule scale. And so it's important to understand that I'm giving you only one small piece of this much broader elephant really in the room that continues to wreak havoc across our global society. And so the fundamental question that we wanted to ask here was, what cells does SARS-CoV-2 target? And so this was really enabled by some of the first biochemical evidence shown here, which I think now is maybe one of the most highly cited papers ever, likely, that SARS-CoV-2 cell entry depends on ACE2 and TMPRSS2. And so as soon as we saw this gave us a hook to look within our data sets for where ACE2 and TMPRSS2 are expressed, if these are, in fact, the receptors that are really critically responsible. And so what this looks like on a slide here is, if we look at the human target cells up at the top and the virus schematized here just as its spike protein on the bottom, the spike protein can dock onto the receptor ACE2. It's activated by a host protease. Its spike protein is activated by TMPRSS2, and then it mediates fusion, a fusion event, either at the cell surface or potentially by endocytosis. These mechanisms are still debated, and we don't exactly know in the relevant target cells which ones are at play. But simply here, we're focusing on how ACE2 and TMPRSS2 might be distributed within these single cell atlases that we've generated. And so here, taking a look at the nasal mucosa, the lung, and the small intestine, we gathered all of our single cell data sets within the lab and worked together actually across really a global set of laboratories in order to validate and extend our results. And we identified that within lungs, we could see just a few of these black points here, marking ACE2 expressing cells. Now, this didn't necessarily look terribly convincing to us. And we think, well, how can such a pervasive and damaging virus rely on just so few cells to gain entry? But when others also started to report similar findings in completely distinct data sets, we started to be a little bit more I guess confident in what we were seeing. And now a lot of the data has borne out that yes, in fact, type 2 pneumocytes in the lung, which are responsible for surfactant production there and keeping the alveolus open, do seem to be principal targets. Now, in the intestine, within this cloud of cells, the black points were much more obvious. And so here we could see ACE2 and TMPRSS2 effectively co-localizing within absorptive enterocytes. And now several reports have highlighted the fact that the intestine can definitely harbor virus, and perhaps for long periods of time as well. And so returning back to the nasal mucosa, we wanted to understand within the same data set that I was discussing in the first part, where might the virus be infecting. And so again, we found just a few ACE2 and TMPRSS2 double positive cells shown here, but they were marked by recurrent patterns of genes, and for some of the aficionados in the audience, I'm illustrating them here. These are genes that mark secretory goblet cells, and really kind of an inflammatory version as well, marked by these genes here. And so what we wanted to understand is, within this, since we have individuals from healthy to a very type 2 allergic skewed environment, what we basically saw is that these cells, the potential target cells, which here are in blue, is if we look within the healthy inferior turbinate, we can see that they make up quite a large chunk of these samples. Now, if we look within the moderate form of chronic rhinosinusitis, we see that they start to be lost and almost completely gone in polyposis. And so this might be kind of a paradoxical aspect here where type 2 allergic inflammation actually reduces some of the target cells that this virus might go after. Now, of course, this was a completely untested hypothesis. We haven't formally tested this, but the epidemiological data bears out that individuals with allergic inflammation in their lungs, except for maybe some types of asthma, don't seem to be at greater risk and maybe even at milder risk. Now, of course, this could be for, they know that they have a lung condition, so they're, more careful in terms of how they, move about the world. They're maybe more careful at adhering to masks, et cetera. So we don't know if this is either the biological explanation or more of the behavioral explanation behind this, but rather we were intrigued by that nonetheless. And so this work was reported early last year in kind of March, April of 2020. And it was really led together by a team of talented graduate students here, and also a variety of investigators around the world with, without their efforts and contributions, this work simply would not have been possible. And here I wanted to highlight a couple of other papers that were companions to this one and organized through the Human Cell Atlas Lung Biological Network. Of course, Alex was really key and fundamental in catalyzing this work, as a lot of the work had been started in his group as well. And so, what I highlighted here was, the identification of some putative target cells, but what was grossly missing is that, of course, none of these samples were from actual COVID-19 infected patients. So we didn't know if this represented just a guess or if it had any foundation in what was actually happening in patients. Now, of course, a lot has happened in a year. We now have developed vaccines faster than for any other infectious disease before. And there has been tremendous understanding gained in terms of the pathogenesis and the immunological factors that can effectively protect against this type of virus. But there are still many open questions of relevance to COVID-19 and also other respiratory pathogens. And so we wanted to ask precisely which are the in vivo cellular targets and factors that influence disease trajectory. So as far as where our current understanding is now, we understand using some in vitro models, and this is the citations are shown here. This isn't our data, but using in vitro models, we understand that basically almost any cell type could serve as a potential target for SARS-CoV-2, at least in a dish. And some early work carried out last summer from a group in Germany gave us an early glimpse that, yes, it is possible to capture virus within single cell data, but due to the heterogeneity and kind of size of that patient cohort, we wanted to see if we could go a little bit further and ask questions within the nasopharyngeal epithelium and a little bit earlier in disease trajectory. What's also been noted studying responses in peripheral blood, which is, of course, typically much more readily accessible, that the difference between mild to moderate, shown here in blue, and critical COVID in red, is a diminished antiviral type 1 response, shown here by a composite score and also individual factors that give rise to this. And rather than maybe a difference in viral load, either within the nose or within plasma. And so this raised to us the important question of, two questions. What are the direct cellular targets of SARS-CoV-2 infection? And also, do these earliest infected cells dictate that defective interferon response, or at least in some way correlated with what's going on in the periphery? And so, one of the things that as I mentioned was really enabling to this work is that it wasn't the first time that we had tried to profile cells off of what effectively is a small toothpick or brush going into the nasopharyngeum. And so what we could do is we could actually leverage the same nasopharyngeal swabs that are used for viral testing. They don't only have viral RNA, they actually have quite a few host transcripts, and not only host transcripts, but actually cells within them. And so we found out that we could take these swabs, put them in a cell viability cryopreservation media, and then freeze them in one location, ship them up here to Boston, and then capture these single cells, again, using the Sequel platform to understand both the host cell identity and also the SARS-CoV-2 RNA positive cells, or at least that's what we thought we would try to do. And so, again, needless to say, this was a fantastic collaboration with investigators at the University of Mississippi Medical Center, led by Sarah Glover. We have never actually met in person, but she's someone who now, I consider really to be a close friend and colleague in carrying out this work. And it has also tied us closer together with investigators here at Boston Children's, namely Bruce Horwitz. And again, this is a work that has been led by a talented MD PhD student, Carly Ziegler, in Alex Shalick's laboratory at MIT, the Broad and the Reagan Institute. And so a little bit about the cohort. What we tried to do is to kind of balance out the cohort in terms of the disease severity that we would be sampling. So we had 15 control individuals, those individuals that didn't have COVID, or at least detectable COVID at that time. Then we had 14 individuals that were within the mild to moderate. So these are individuals that, of course, are sick enough with COVID to present to the hospital, but they do not need respiratory support that requires mechanical ventilation. And that is unlike those in this 6 to 8 category that are those on more of a external oxygen type of support rather than just something that might be on demand. Here, this is showing a further breakdown of these categories. Again, we're looking at 1 to 5 in aggregate and 6 to 8 in aggregate. And unfortunately, those individuals that have met the criteria for 8 are those that unfortunately passed away from COVID-19. And we also have a few controls within a WHO 7 to 8 criteria that you'll see here in a couple of the slides. And those are intubated controls that didn't have COVID infection. Now, this is a cohort that is representative of the demographics that the University of Mississippi Medical Center serves. And it was actually, at least surprisingly to us, quite balanced across the age range as well. So we're not actually just sampling differences in age susceptibility, for example, in these cohorts. And so, as I mentioned, there was a bit of effort that went into developing some new protocols to really recover every last viable cell from these frozen nasopharyngeal swabs. And so this protocol, I won't belabor the point here, but this protocol is available on protocols.io for those of you who might be interested. And it's schematized graphically here. And this allows us to recover those nasal epithelial cells from these nasal swabs. And so just jumping immediately right into the data, we, across these 58 eventual individuals, we recovered 32,500 single cells total. And what I'll do over the next couple of slides is orient you as to who these cells are and then how they're changing during disease. So here I'm circling with my mouse the epithelial cell compartment. So starting from the basal progenitor cells, those stem cells, that then through developing secretory and goblet cells give rise to goblet cells and secretory cells. There's this deuterosomal cell cluster that actually is a transient intermediate on its way to developing ciliated cells and eventually to ciliated cells that are, again, those that help to sweep away agents that are found on the epithelial surface. Now here, there's also, of course, because they are not just nasal, but rather nasopharyngeal swabs, there's also squamous cells that we're sampling that are from a deeper region, and then immune cells like T cells and macrophages. And then also here, this cluster of ionocytes, for example, that was recovered and actually quite a recently described cell type through single cell data in mice. And so here we can actually recover this and it clusters quite nicely. And so now what I'm doing is I'm showing you the same single cell representation on the left. And now this is colored by not infected versus non-infected cells, but rather the SARS-CoV-2 PCR diagnostic assay for each patient. And we can immediately see that certain areas are overlapping, showing both the green and the red. And then there's some areas that are predominantly composed of only the red cells. Now what we can do is we can start to perform just simple statistical tests to understand which of these cell clusters are over or underrepresented based on disease severity. And so we can see that, goblet cells are not significant, but some patients start to be kind of outliers here. Secretory cells are significantly increased with COVID severity. Deuterosomal cells are increased, developing ciliated cells are increased, and overall, those mature ciliated cells are significantly decreased. Now, what, since I introduced the concept of epithelial diversity, we also wanted to ask here, what is going on with the overall epithelial diversity in COVID-19? And now we see, unlike allergic inflammation, where you have a suppression, at least over this short time scale of COVID-19, we have an increase in epithelial diversity, as it seems like patients are starting to explore a variety of different cell subsets that normally don't exist. So this might be distinct from some of the inflammatory or chronic inflammatory diseases. Now, the epithelial barrier, of course, is a dynamically differentiating tissue. And so we wanted to move away from just a static type of snapshot and rather incorporate some of the dynamics of RNA metabolism. And so using a series of techniques under the umbrella of something known as RNA velocity, we were able to basically leverage the transcriptional on rate, the splicing rate, and also the RNA degradation rate in order to ask, effectively, can we draw arrows through that plot so that we can then see which way these cells are differentiating? So this is what we tried to do. What I'll show you on the next slide is on the left, the samples from our controls, and then on the right, those from COVID-19 positive individuals. And so we can see here the basal cells now in the bottom right, going up towards secretory and goblet, and then they basically just stop there. Then there's kind of like a jump here that appears to be absent in a healthy epithelium, where we go towards the developing ciliated and then eventually mature ciliated cells over in this direction. Then there's the squamous cells here that, as we suggested, are kind of their own island. And now what we can see is that in disease, now these secretory and goblet cells, some of them are actually going backwards, seemingly de-differentiating, while other ones are jumping over towards these deuterosomal cells and towards these developing ciliated cells and pathfinding their way towards the more mature ciliated cells if they can make it. And so it seems like there's both ciliated cell replenishment and also de-differentiation events that are ongoing among secretory cells. And so again, returning back to the kind of discrete compositional aspects and looking at all these fine grain clusters that we identified, we could see that there are significant increases in secretory cells in a variety of these subsets, again, with disease severity. That goblet cells, while none of these are significant, certain patients start to increase their numbers of certain types of goblet cells. And a couple of these here on the right will become important in a moment. And then the ciliated cells are significantly decreasing in general, especially those that are the most mature and stable subsets. And that's summarized down here. So of course, we weren't able to just go in and say who these cells are, but rather we wanted to ask, on a gene by gene level, once we've been able to bin them out, how different are they in terms of their differentially expressed genes within each relevant comparison. So what I'm showing here is the entirety of the different fine-grained cell clusters that we identified in the data set. And what we're doing is we're basically doing 3 differential expression tests. We're doing one between control individuals and those with mild to moderate. We're doing control to severe and then mild to moderate to severe. And immediately here we can see that not all cell types are equally impacted, and there's some that have more significant changes than others. And so I'll focus in on one of these that happen to captivate our attention, and that's the ciliated cells and how they are changing their expression with disease severity. And so here, what we can find is a variety of genes that are significantly increased in ciliated cells with individuals with mild to moderate disease. And these are really canonical interferon-induced, so type 1 antiviral genes. We see kind of the same, but slightly lower expression of some of these in the severe. And what's really highlighted, if we compare mild to moderate versus severe, those severe individuals have almost a complete muting of their type 1 interferon response. And so, of course, we wanted to ask, is this not only happening in ciliated cells, is this more broadly distributed? And the answer is that yes, this is a global signature across several epithelial cell subsets shown here. So the middle one in red is the mild to moderate, and the one in the lighter red color is the more severe one. And we can see that this is significantly increased in almost all of the comparisons. And so, of course, the next logical question after looking at something that is responsive to interferon is who is making interferon. And this is where some of our results, at least for the time being, are still somewhat inconclusive. And so if we look here, again, at all the clusters and all the different type 1 and type 2 and type 3 interferon upstream genes, we can find type 2 interferon gamma, made by T cells, as we would expect, but absolutely nothing here that we could detect. And so that, of course, always raises the question, as I brought up, that single cell is not necessarily sampling the totality of transcripts, and interferons can be challenging to detect for a variety of reasons. And so is it simply that our platform isn't capable of detecting this, or is it that this actually reflects relevant disease biology? Now, in order to ask this question, we could rely on a study that we performed with the same platform on the 2017-18 Massachusetts flu season. And here we can actually pick up both interferon alpha and beta, and actually interferon lambda expression as well in influenza, showing us that we can pick it up in ciliated cells, as I'm circling here, in virally infected cells. And so this to us highlights that it's not necessarily platform, but perhaps representative of biology. And I think some of the in vitro work is also supporting this. Now, for now, we focused on the host changes in both cellular composition and individual RNA molecules. But what we also wanted to ask is, can we find which are the virally infected cells? And so what we did is we went in and we initially asked, well, can we find any types of viruses in here? And so here's all of our different participants along this axis, and moving up this way across all the rows, we have a variety of different common respiratory viruses. And we can see that within the severe and mild to moderate, we're aligning to SARS-related coronavirus, so presumably SARS-CoV-2. And that within some individuals, we're actually incidentally picking up things like rhinovirus or enterovirus, or even blips of influenza. Of course, this isn't a clinically validated assay, but interesting to consider nonetheless what some of these might mean. And even some of the control individuals had little blips of virus in them as well. But what was important to us is that we could reliably detect these SARS-CoV-2 reads within most of our COVID-19 participants. So emboldened by this, we went forward with trying to ask which cells are actually infected. And so, of course, we can look at the totality of SARS-CoV-2 transcripts per cell, shown here, again, kind of quantifying a little bit more cleanly what I showed on the last slide on either a per sample or on a per cell level. Now, what we can also do, in addition to just looking on this level, is we can actually look for all the different viral life cycle intermediates in terms of RNA. So SARS-CoV-2 is a positive sense single-stranded RNA virus, and we can detect both the positive sense, and we can also detect the negative sense RNA intermediates, which are important templates for making more positive containing virions, as well as nested subgenomic RNAs that are basically this leader sequence fused to a variety of the other sequences, S, M, E, etc. And so what we did is we went forward, we applied a background correction algorithm to correct for ambient viral RNA correction, and then we looked for basically where these points here are in terms of all the cells that we had already described. And so what we came up with is basically this, that by detecting these RNA viruses, we can find these RNA positive cell types within a variety of different infected individuals. So we can find keratin 13-24 high secretory cells as prime viral targets, these AZGP1 high goblet cells, these CCL5 high squamous cells, developing ciliated cells, and also interferon responsive ciliated cells. So of course, now the question you might have is, well, we had a study that came out last year on putative infected cell subsets. Were we in any way, shape, or form close to what we thought might be the case? So here, going left to right, the higher this is, the closer it was to our predicted primary cell target. And if you focus on this part of the plot, what I'm showing you here is the frequency of that cell cluster that's expressing either the positive sense, the negative sense, the unspliced or the spliced variants of the SARS-CoV-2 genome. And so here, focusing in on this one, which is one of our primary candidates, we can see that these are quite high in terms of the frequency of infected cells, these AZGP1 high goblet cells. Same for these keratin 24-13 high secretory cells. And then what was interesting is that also some of them that may not have been found within the samples that we were studying are also prime targets, like these interferon responsive ciliated cells, for example. And so another important point to make here is that, we also have immune cells, not only epithelial cells. And so within these macrophages, we can also see that macrophages are capable of accumulating virus. However, they have a distinct distribution of this kind of replication stage. And so, of course, the question here, is it engulfing free virus? Is it engulfing these infected cells? And what is the mechanism of infection of a macrophage relative to an epithelial cell? And that's highlighted a little bit better on this plot here, where if we look at the percent of ACE2 positive cells going along on this x-axis and compare it to the percentage of SARS-CoV-2 RNA positive cells on the y-axis, we can see that the goblet cells are correlating quite nicely, but that the interferon responsive macrophages are off of this diagonal, suggesting that the virus is not getting in through a canonical ACE2 mediated mechanism. Now, the last question that we wanted to ask, at least within the work to date, was, if we can now look at viral targets, can we then separate those cells that are coming from an infected tissue ecosystem from those that are not infected? Now, of course, they may have been or are about to be infected, but at the time of sampling, they were not infected. And is there anything that is significantly distinct between these two different classes? And so what we did is we performed differential expression across all of these and came up with some recurrent signatures. And what we found consistently higher in bystander cells were these MHC2 components that are strongly upregulated. And actually a close friend and colleague, Samir Beyaz, has pioneered work on MHC2 in the intestinal epithelium. And it would be great to learn more from him and understand a little bit better about what this might mean for viral susceptibility in this context. And so to summarize here and to conclude the talk and hopefully open up for any questions if time allows, what we found is that impaired local intrinsic immunity to SARS-CoV-2 infection is likely to underlie severe COVID-19. We've generated an open resource of COVID-19 positive nasopharyngeal swabs. The cell atlas is available here at this link. And there's also actually a series of other atlases that are available from other tissues that were generated through a CZI single cell COVID-19 consortium. We've identified the full epithelial trajectory from these frozen swabs. We've identified changes in epithelial composition. The SARS-CoV-2 positive and negative strand seems to be highest within goblet and secretory cells. And as I mentioned at the start, that severe COVID-19 appears to be characterized by this muted interferon signature in the initial barrier tissue targeted by SARS-CoV-2. And so in order to wrap up, what I'd like to do is place this into the overall context of what our current understanding is. So of course, we're not the only group by any means studying this. And so there's quite a lot of activity happening in the SARS-CoV-2 and COVID-19 space. And so I wanted to highlight a couple of other of the pioneering papers that have identified this reduced interferon stimulated gene signature either in PBMCs or even identified upstream mechanisms, whether it be genetics or autoantibodies that could be leading to this muted and blunted type 1 interferon response. And I think that there's a lot more to do here to understand whether these mechanisms are also underpinning what we see. And so the current context in terms of what we understand a little bit more globally is that interferon is important, but timing and location of interferon is even more important. So there's a few papers from either SARS-CoV-1 in mice or SARS-CoV-2 in hamsters suggesting that interferon does have a bit of that double-edged sword phenomenon going onto it. And so this is highlighted here. And there was also some really nice work from Ivan Zanoni's group looking at how type 3 interferon lambda might actually be detrimental. So it could be beneficial in one location, but it could be detrimental, at least in certain forms within the lung epithelium as well. So there is further complexity to appreciate here within the system. And so appreciating the impact of SARS-CoV-2 on systems overall is, of course, going to take more than just what I've been able to share with you today, where we focus more on cells, barrier tissues, and genes. But there's been really pioneering and fundamental work on the viral genomic elements themselves, on virions, and on molecular host-viral interactions. And of course, we seek to understand how all of this feeds into, intertissue organismal levels of communication, what this means actually for people, right? In the same way as we have kind of super spreader cells, and super spreader people, are these correlated in some way, and what these means overall for our communities. And so circling back to this idea that, immune cells and tissues probably mean more than just the immune cells that we think about. Epithelial cells, stromal cells, even neurons are really critically involved in host defense, both protectively and also potentially detrimentally in states of inflammatory disease. And I think that this is really an exciting area going forward. And so what, just on the last slide, what we're doing in the lab now is building out a variety of these different human single cell atlases building on some of our published and ongoing work in the intestine, in the skin, and the airway, in order to identify states that seem to be important in those diseases, and then really asking, once we've observed these cell states, how can we really go on and determine their role in either protective immunity or detrimental inflammation. And so with that, I'd like to conclude by acknowledging everyone that made this work possible, in particular the more recent COVID work that I've shown. And of course, this was a huge team effort, both from my postdoctoral lab, Alex Shalik, who's been a fantastic and incredibly supportive mentor, new friends and colleagues at Children's, friends and colleagues at Brigham and Women's, and also at University of Mississippi Medical Center. And here really showing the ICU nurses that help make the latter part of the work that I've shared with you possible. And so with that, I'll conclude here. I'll thank all of my lab members and also funding sources, and leave my email here in case anyone has any questions. So thank you very much for the opportunity, and happy to take questions.
Thank you so much for this very interesting, comprehensive, and exciting talk.
You're very welcome. Thank you.
Please ask questions which can be asked, send a message through the chat button to the speaker or to everyone, or just use a microphone. So questions, please.
I'm monitoring the chat as well in case anyone has anything there that they'd like to ask. And I'm also happy to discuss any general aspects or interest of COVID-19 as people might be curious.
Can you see chat?
Yeah, I can. Yeah, I can.
Okay.
Apparently the issue is that there's unmuting issues. If you like, you can type into the chat quickly, or maybe it's possible to unmute folks individually as well.
I think I managed to unmute. Thank you.
There you go. Yeah.
Thank you, Jose. This beautiful presentation. I'm not a biologist. I'm a physicist from Argonne National Lab. My question is, after listening to all these diagrams and things that I don't understand, how far you are in terms of developing treatments?
Yeah, so I mean, I think there's a couple things here, right? So treatments, obviously, vaccines are front and center, right? And we've had absolutely nothing to do in terms of the vaccine development. That has really been pioneering efforts by many dozens of groups around the world. But what I can say regarding the vaccines is that there's I think really important unanswered questions regarding protection from severe disease, which of course is paramount in terms of getting out of this pandemic, but also thinking for other respiratory viruses, SARS-CoV-2 included, what and how these vaccines can be optimized to provide protection not only for myself, but also to really optimally protect for transmission as well. And so this is where vaccines that are not only systemically targeting, but actually targeting some of the primary tissues that are infected are going to be really important. So we're interested in some of those questions moving forward. In parallel to that, of course, there's treatments that could be considered either as kind of simple prophylactic treatments, say we don't have a vaccine yet, how might you protect or bridge until you have a vaccine? One simple but challenging thing that has been proposed is the use of prophylactic type 1 inhaled interferon, right? But we also don't know the chronic long-lasting effects of some type of treatment like this. Is it something that someone can do for a few days, a week, a month? What is that going to do to the tissue? And it's still a recombinant protein, and so there's certain limitations to how scalable that is worldwide. And so I think that those are two examples where there are currently therapies in existence, but they're based on some of the lessons that we're learning, we can perhaps go in and hopefully optimize some of those aspects, especially within the nasal epithelium.
May I follow up with another question if there is nothing else?
Yeah, go ahead. I see that there's a couple more coming in, but go ahead. Well,
excellent. So here at Argonne, we solve a lot of protein structures, structural biology work with respect to COVID-19. How relevant those studies are in terms of guiding your work?
I mean, I think it's really of critical importance. So one of the things that we've been looking into is not only how to measure some of these things in RNA space, right, where it's not necessarily an immediate one-to-one with protein, but rather, if we can go in and use different, protein probes designed by the virus as kind of molecular reagents to be able to probe for these cells within samples, right? So absolutely, that is very important. And so having a molecular understanding of the host viral interface is essential to design any of those reagents. So absolutely, I think it's very important.
Thank you.
You got it. I also saw that there was a question, I'll just read it here. I was just curious that you stated that COVID-19 infection actually causes stagnation in cell differentiation. So what I mentioned as far as the cell differentiation was more under the allergic inflammation context rather than COVID-19, where those cells appear to be getting stuck in terms of the ability of them differentiating towards secretory cells. What we're seeing in COVID-19 is that actually now secretory cells may go from 2 states that are present in health to 6 or 7 different ones that are present in disease. And so it seems like there's an exploration of multiple cell states. It's possible that, if I can paraphrase, you might have been referring to also the de-differentiation effects. This is something that several labs have been studying quite actively. I think is a really exciting question about the plasticity of these epithelial barriers and how we can, typically might think about them as a one-way road, but these cells are probably filling in different gaps within the epithelium as necessary. And so infection might be causing certain gaps that these cells are effectively trying to inhabit. So I think it's a good question. There's also another question on, do you think these studies could pave the way for developing inhaled vaccines? So there's some inhaled vaccines efforts going on now already. And so I think that, of course, being able to pair the types of analyses that we do on inhaled vaccines would be really fascinating and interesting. I would love to do something like that. So yes, I think that in flu and in COVID, that also is a very important point, and what I was alluding to in the first question. And then the last question that I see here is it possible to prevent the entry of virus into the cell by inhibiting clathrin-coated or caveolar vesicle activity? So it's not a focus of our group, but I've had a conversation with Tom Kirkhausen's lab, who's here at Boston Children's as well, who actually asked me that question during a seminar as well. And I think his group has been following up more specifically on the mechanisms of endocytosis. As I mentioned, it's still an active area of investigation, so I don't want to comment incorrectly on what the latest data shows. But definitely check out preprint servers, because I think there hopefully is something soon there from either his group or others that have focused expertise on that area. So I think those are the questions that I saw so far. And then yes, so that's a good question. I think the control patients, it's unlikely that those would be super spreaders per se. I think the super spreaders are probably coming more from the COVID-19 infected or bona fide infected individuals. We understand that those individuals that have a lower CT value from the RT-PCR and thus a higher amount of viral RNA positive are probably those that are spreading most effectively. Actually, some of those very low values may not be able to, high CT values, low viral values may not be able to spread so efficiently. And so the question, I think, is rather, which are the ones within the mild to moderate and severe that actually do appear to kind of behave as super spreaders? This we don't understand. I would love to do this at a epidemiological scale if funding were to allow, but an important question that you asked. Okay. So as I said, if there's any further questions, please email me, email us, find us on our website or on Twitter. Yeah, I'll just say thank you very much again for the opportunity to be here, and hopefully someday I would love to visit Turkey. I haven't been able to be there yet, so looking forward to the opportunity if it presents.
Yeah, sure. Thank you so much. That was a big pleasure for us. We appreciate it very much. And
yeah,
I also hope that we will see you in person in the nearest future, as soon as the COVID outbreak ends, finally. So take care of you.
Yeah, thank you very much.